Okay, Assalamu alaikum wa rahmatullahi wa barakatuh. My name is Samhar Alouch. I'm a second year medical student. And today we have one of the most important lectures in the whole MOL thing, which is cell signaling 1, 2, 3. I will be explaining cell signaling 1, 2, and my friend Amjad will explain cell signaling 3. So without any further ado, let's get started. One thing I want to mention is that I'm using Dr. Ahmed Al-Jada slides. And I did a little modification. It's not that important. And I highlighted things with yellow. That's just for me. Other than that, it's the most important things. But I literally, most likely highlighted most of the things. So, rely on the lecture and please focus on the explanation because this lecture needs understanding more than memorization. There is memorization part, which is the pathway, but focus mostly on understanding the concepts, okay? Because the questions are mostly from understanding. So without any further ado, let's start. So first we will start with cell signaling 1. So yeah. And also I will be going back and forth into this white sheet to explain on it, then we go on the slides. So let's start. First, you learned in the whole blocks that you took that it's like this. We have the membrane of the cell, and on the membrane, we have a receptor. On the receptor, a ligand bind or a voltage-gated channel or many things, but this is a channel. When the ligand binds, it opens and it allows ions to go in. Or it has another way or another pathway, which we will explain today. Okay, that's just saying. So we have different types of cell-cell communication. We have cells in our body and the cells communicate with each other. Like they send signals to tell each other what to do, okay? And we have 4 different types of communication. The first one is direct communication. Direct from its name, that the cell is directly attaching into the other cell. So it directly communicate with it. There is no signals, it's literally touch it. Okay, so one cell have molecules like this, and the other cell have receptors. The first cell go and bind into the second cell, and it attach to it, and that's the direct attaching. This is the first one, direct contact. And it's, as I said, molecules on the surface of one cell recognized by receptors on the adjacent cell. And it's important in early development, and there are gap junctions in it, okay? So this is the first type. The second type is paracrine signaling. It's like this. We have one cell and the other cell. The first cell sends signals locally, okay? So to things that are near it, okay? So it sends the signals and molecules, and those molecules go and bind on their receptors. So the two cells doesn't touch each other. The first cell sends molecules and they bind on the second cell, and that's it. And because of this binding, channels open or internal signaling pathway happen. So this is the second type, which is paracrine signaling. The first cell sends molecules to other cells locally, but they don't touch each other. The two cells doesn't touch each other, but the molecules go all the way and bind to the second cell receptor. And that's the second type. Okay, just a second. Okay, so as I said, signal released from a cell has an effect on neighboring cell. And it's also important in early development, and it coordinates many neighboring cells, and it is important in signaling of immune cell. And that can be an MCQ, like which of the following signaling is happen in the immune cells, and it's paracrine signaling. The third way is the endocrine signaling, and it's like this. We have the blood vessel. Okay, this is a blood vessel. And the first cell sends its molecules into the blood, okay? And then these molecules go with the blood, and then from the blood, it goes and binds to another cell. So it sends the signals like the paracrine into another cell. It doesn't actually attach to it. It just sends the molecules, but it doesn't send it locally. It sends it to the blood, and from the blood, it goes all the way and go and bind to another cell. Okay, that's the third type, which is endocrine. It's like hormones. It goes from one cell into the blood, then from the blood into the other cell. Okay, so this is the third type. Hormones released from cell travel through circulatory system, which is blood system, to affect another cell. and it happens in animals and plants, and they use this mechanism most of the time. Third mechanism is synaptic signaling, and from the word synaptic, it means that we have neurons, and you took this in MSK. Okay, so we have one axon. Okay, this is a neuron cell, and the axon have axon terminal at the end, and it sends the signaling molecule, and it binds into the muscle or into another neuron or anything else. Okay, it involves neurons. So this is the fourth type, which is synaptic signaling. So it happens in animals. It doesn't happen in plants, only animals and the humans. And nerve release the signal and binds to receptors, and it's associated with the neurons and the target cell. And the place that the synaptic neuron and the cell have the molecules bind is the chemical synapse. Is everything clear? This is just a recap. I'm sure you took this somewhere else. So is everything clear? Okay, so let's continue. Now we will talk about the actual content, okay, which is the signaling pathways. So let's continue. So now normally, as I said, we have the membrane of the cell and we have the receptor. This is the receptor. So in the signaling pathway, we have 4 components. First, we have the ligand, okay? Ligand is the molecule that go and bind into this receptor. So now it binds into the receptor. The second component is the receptor. So first we have the ligand, which is the molecule that binds into the receptor. The second thing is the receptor, which binds to the ligand. Now, after the binding of the ligand, this receptor activates an internal signaling pathway, like it activates many enzymes under it, or it just opens. When it opens, it allows ions like sodium, potassium, or whatever to enter. And now they enter, and they activate internal signaling pathway or whatever. So first, signaling is the cell-cell communication. Okay, so cell-cell communication happen through signaling, which is molecule bind into the receptor and the receptor open, allow ions to enter, or we have something called internal signaling pathway. So, signal transduction, it's the process of converting extracellular signal into intracellular response. Like the binding of the molecule is causing a phosphorylation into a specific protein, or a binding of the molecule causing calcium to increase inside by opening the ions. So signal transduction is converting the extracellular signal, which is the binding of the molecule, into intracellular response, which is activation of a specific enzyme or phosphorylation or opening of a channel. So that's what signal transduction means. Ligand is the signaling molecule, and receptor is the thing that binds to the specific ligand. So the components are ligand, receptor, and the intrasignaling proteins. And these are the things like enzyme, second messenger, target proteins, many other things, okay? So this diagram is actually cool. We have the molecule go and bind into the receptor. The receptor activates internal signaling pathway, which means that it activates one enzyme. This enzyme activates another enzyme. This second enzyme activates a third enzyme. And this third enzyme activates a fourth enzyme, and it continues till it reaches to an enzyme that causes a response, okay? And one important thing to mention that we have 2 types of signaling, okay? The first type is that after the ligand binds to the receptor opens and it allows ions to enter and exit, okay? That's the first type, which is the general type, okay? The second type, which mostly in this lecture, is that after the ligand bind into the receptor, it activates internal enzymes. So it activates an enzyme, and this enzyme activates another enzyme, and the second enzyme activates a third enzyme, and we continue till we activate something that causes a response, okay? So we have 2 types. First, the one that opens the channels, causing ion to go in and out. And the second one is that after the ligand bind, it activates internal signaling pathway or intracellular signaling pathway, which is an enzyme activates another enzyme, activating another enzyme till we reach a response. It's important to mention that the one that has ions to open, after it binds, the channels open and the ions enter and get out. That's it. This one, since it has many enzymes activating each other, the signaling response is get amplified. So if like one molecule bind here, the effect will be very much high. Why? Because this enzyme is actually activating another enzyme, and this way, by this cascade, or this thing, like enzyme activating another enzyme, activating another, it's actually causing an amplification of the response. So actually, at the end, we will have a very big response. Not like here, if one molecule bind, it will open the channel, causing like one ion to enter, and that's it. But here, after the ligand bind, it will cause internal signaling pathway that cause many enzymes to get activated, and it causes something called amplification of the response. So one molecule may cause a very big response. Okay, is it clear? If it's not, please interrupt and tell me that, oh, we didn't understand. And please write on the chat. Okay, clear. So let's continue. As I said, we have receptors in our body. These receptors can be on the membrane of the cell. Let's say this is the cell, okay? And this is the membrane. We may have actually the receptor on the membrane. So the ligand go and bind into the receptor, and that's it, because the receptor is on the membrane. The other type is intracellular receptor. So the receptor is actually inside the cell or inside the nucleus. Okay, so the ligand need first to pass the membrane, then bind into the ligand, which is nuclear receptors or intracellular receptor. So we have two types of receptors. First one is membrane receptor, where ligand bind into the receptor on the membrane. The other type is nuclear receptor or intracellular receptor, and the receptor is inside the cell. So ligand first need to pass by the membrane and then bind into the receptor inside the cell. And so the membrane receptors, usually the ligand is low concentration. Why? Because as I said, mostly in membrane receptors, they use the other type of signaling, which is the cascade, which has amplification. So low concentration actually cause a big response. Why? Because usually the ligand is low in concentration, and transduction event has signal amplification. So we must memorize that membrane receptors, which is on the membrane, low concentration of ions can cause low concentration of ligands can actually cause big response. Why? Because they depend on the signal amplification. And usually those ligands that bind on the receptor are usually peptides and hydrophilic, which means that they can't pass by the membrane, so they can't bind to intracellular receptor. They can only bind into membrane receptor. So after they bind, they cause signal transduction, which means an enzyme activates another enzyme, activating another enzyme, and by that, they cause amplification. So low concentration can cause a big response. The other type is nuclear receptor, which means that the receptor is not on the membrane. Actually, the receptor is inside. So first, the ligand need to pass by the membrane. And to pass by the membrane, the receptor must be, the ligand must be hydrophobic. Okay, so again, in the nuclear receptor, the receptor is inside the cell or inside the nucleus. So the ligand must pass by the membrane to actually bind to the receptor. And to be able to pass by the membrane, the ligand must be hydrophobic. So the ligand for nuclear receptors are hydrophobic because they can pass by the membrane. And so they bind into the nuclear receptor or intracellular receptor, and they cause the response. And this response has no signal amplification because it just open the channels and cause ions to enter into the nucleus, or it just go to the nucleus and bind into the DNA. So it doesn't have this cascade of enzyme, like enzyme activates another enzyme, activating another enzyme. No, that doesn't happen in the nuclear receptors. Nuclear receptors just bind to the ligand, then they cause the response, okay? And the response is direct. So there is no signal amplification. And since there is no signal amplification, we need a very high concentration to cause a very high response. So last time I will say this, just a second. Yeah. we have two types of receptor. First, membrane receptor, it binds to the ligand outside. So the ligand can't pass by the membrane because it's hydrophilic or peptide. And so it binds to the membrane receptor and it causes amplification because it activates an enzyme, which activates another enzyme. It has this cascade. So it causes amplification, which means that even if we have low concentration of the ligand, we will have a very big response because of the amplification. The other type is nuclear receptor, which is inside the cell. So first, the ligand must pass by the membrane because the ligand must be hydrophobic to pass by the membrane, and then it binds into intracellular receptor or the nuclear receptor, and it causes the response directly. So there is no amplification, which means that we need a very high concentration of the ligand to cause a very big response. Is it clear? This is very important because they bring question on it a lot. If it's clear, please write yes. If it's not, please write no. Oh, you can't open mics? Sorry. Okay. So is it clear? So shall I continue or someone wants me to repeat this?
Can you repeat this, please? It's kind of hard.
Okay, we have two types of receptors. First one is membranous receptor, which is on the membrane. So the receptor is on the membrane. And since it's on the membrane, the ligand binds on the membrane. It doesn't need to pass by. Or actually, the ligand can't pass by the membrane. It must bind here. And thus, the ligand is hydrophilic or peptide or amino acid, which can't pass by the membrane, okay? So they must bind to the membranous receptor. And once they bind, the membranous receptor usually have the enzyme cascade. So it activates one enzyme, which activates another enzyme, which activates a third enzyme, and we keep continue till we have the response. And this cascade causes amplification. What does that mean? It means that if we have low concentration here, it's still okay, because low concentration of ligand cause, with the amplification, cause a very big response. Because imagine we have two. With the amplification, it become 4, 8, 16, 32, and we keep continue till it become big, and we have a very big response. But in the nuclear receptor, this is the nucleus, let's say, or this is the cell, and this is the membrane. And so the receptor is here. So the ligand need to pass by the membrane to bind the receptor. And to pass by the membrane, the ligand must be hydrophobic. Okay? So first, for nuclear receptors or intracellular receptor, the ligand is hydrophobic. And once it binds, it doesn't activate many enzymes. It causes the action directly, okay? And since it causes the action directly, there is no amplification. So low concentration of ion or the ligand cause low response. So we need a very high concentration of the ligand to cause a high response in the nuclear receptor. Is it clear?
Yeah, I just have one question.
Go ahead.
On the nuclear receptors, does like the ligands have many receptors that they bind to, so the amplifications be better? I mean,
what do you mean?
I mean, does the ligand connect to many receptors, or just the concentration of ligands it needs to be higher? Does the receptor concentration has to be high too?
No, the receptor shouldn't be high because the receptors are always there. Okay, so ligands just must come and bind because they bind, cause the reaction directly, and then they leave. Another ligand come, then it leave, then another ligand come. So we don't need very high concentration of the receptor, but we need a very high concentration of the ligand because the ligand cause the reaction directly, then leave. So another one can bind, adi. We don't need very high concentration of the receptor, no.
Yeah, okay, it's clear, thanks.
Okay, so now let's continue. Extracellular signaling molecules. So now we said that we have, by the way, this first two lectures, which I will explain, will be about membranous receptors. My friend will actually explain the steroids receptor and nucleus receptors and these things. So from now on, everything will be about membranous receptor, okay? So let's start. Now we have membranous receptor. So this is the membrane and this is the receptor. And as we said, a ligand binds. This ligand that binds, we have 4 types of ligands, okay, that binds on membranous receptor. The first one is small molecules, which is amino acids and its derivatives like glutamate, glycine, acetylcholine, and these things. So this is the first type of ligands that binds on membranous receptor. The second type is steroids, which is derivative from cholesterol, let's say. Eicosanoids, which are from arachidonic acid, which is fatty acids. And finally, we have polypeptides and proteins. This is just memorization. You have nothing to understand from here. So, and that's for the ligands, okay? So ligands come and bind into these receptors, membranous receptors, and some of them actually can bind on membranous and nuclear. But these 4 can bind on membranous receptors, okay? These are the ligands, okay? Now this slide is about the receptors. Also, since we have 4 types of ligands that can bind into receptors, we have 5 types of receptors which can bind to these ligands. We actually will focus on 4 only. So the first one is G-protein coupled receptor. What does G-protein coupled receptor mean? We will explain it in detail, but just for this slide, it's like this is the receptor, and it has a G-protein coupled to it inside the cell. So it's a receptor that has G-protein glued to it, okay? This is the G-protein coupled receptor. It's a receptor that has a G-protein glued to it, okay? Or attached to it. This is the first type, okay? Second type is receptor protein tyrosine kinase. It means that we have the membrane, and this is the receptor, and inside the receptor, there is a tyrosine kinase. Kinase means that it's something that causes phosphorylation, that binds a phosphate into the thing. So if we have a kinase and it goes and binds to enzyme, it will cause the enzyme to have a phospho group on it, okay? So that's the action of kinase. So the second type of receptor is receptor tyrosine kinase. It means that the receptor itself has a kinase, okay, here. So once the ligand binds, this kinase gets activated and it phosphorylates many enzymes inside. This is the second type. The third type is the normal type, which is ligand-gated channels. It means that we have a channel and the ligand bind to it, and then the channel open and ions can get in and out. This is the third type. And fourth type is steroid hormone receptors, which is, my friend will explain it in detail, but it's like ligand bind into receptor adenylate. And fifth type is unique mechanisms, which doctor said don't study it, so it's not important. So is these 2 slides clear? Just an intro into the type of receptors. We will take in detail each one. Is everything clear so far?
Yeah.
Okay. So now, this is the last time I will mention the type of receptors. First one, we have chemically gated ion channels, which means that the ion bind into the receptor, then it opens it and allow ions to enter and get out. The second type is enzymatic receptor. So it means that the ligand or the ligand binds into the receptor and activates an enzyme inside. And this enzyme usually is a kinase, which phosphorylates many enzymes inside. So the receptor itself has a kinase, which means that when the ligand binds to the receptor, it activates this kinase and it causes phosphorylation of many enzymes. The third type is G-protein coupled receptor, And these are a receptor that has G-protein attached to it. It's like a receptor, and we have G-protein attached to it. Once the ligand bind, the G-protein detach and go away, okay, and do its action. And the fourth type is the steroid, which my friend explained clearly. This is the first checkpoint. So everything so far, the whole thing is clear till now, because we will start with deep things now and important things. Everything is clear?
Yeah, so far everything's good.
Okay, so let's continue. So we will start first by G protein coupled receptors. And as I said now, G protein coupled receptor is a receptor that has a G protein attached to it. I will ignore the slides completely now. I will just draw and explain. Please pay attention and understand. Then at the end, we'll go to the slides again. So first, as I said, we have the receptor. Let me change the color. We have the membrane. And then we have the G protein coupled receptor. How does it look like? The G protein coupled receptor has a very special structure, which is we have 7 helical structure, and helical means it's like this. So we have 7 helicals, which are like this. 7 helicals. These are 6. This is the 7th. 7 helicals. Okay, this is alone is the receptor. 7 helicals, and it has an N terminal here and a C terminal here. And above we have connections like this. So it has 7 helicals. This alone, this whole thing is the G protein coupled receptor. It has 7 helicals. It has an N terminal out of the membrane and a C terminal inside the cell. Okay, and it has 3 extracellular bridges and 3 intracellular bridges. Okay, this is the structure. 7 helicals with N terminal outside and C terminal inside. And we have 3 bridges outside and 3 bridges inside. Now, important points to mention that these bridges has disulfide bond. He can ask you, what is the type of bond that keep these bridges together? And it is a disulfide bond. This is the first important thing. Second thing, it has the N terminal outside, and the N terminal, since it's outside, it's the ligand binding site. So when the ligand will bind, it will bind to the N terminal, okay, because it's outside. And as I said, it's a receptor that has a G protein attached to it. So G protein must be attached to this receptor. Where does it attach? It attaches at the C terminal. So here we have the G protein attached to the C receptor. Okay, so this is the structure, 7 helicals, N terminal outside, which is the ligand binding site, and the C terminal is inside, and it's the G protein binding site. And we have 3 bridges outside, 3 bridges inside, and each bridge is made of disulfide bond. This is the structure. Now, what is the name of this structure? Since it has 7 helicals, we can name it with different names. First, we can call it G protein coupled receptor because it's a receptor that has G protein coupled to it. So the first name and the most common name is G protein coupled receptor. Okay. The second name is 7 transmembrane receptors because it has 7 helicals that are passing through the membrane. So 7 transmembrane receptors. Third thing is heptahelical. Hepta means 7. Helical is the helical structure. And since it has 7 helical structure, it's called heptahelical receptor. And the last thing is serpentine receptor, which is coming from the word serpent, which is like a snake. So it looks like a snake. So it's called serpentine receptor. It's very important to know each name because you can use all the names in the exam, okay? So please know the 4 names: G protein coupled receptor, 7 transmembrane, heptahelical, and serpentine from the word serpent. Is it clear? The structure. Okay, so now let's take some details about it. It's 7 transmembrane receptor. It is the largest, most ubiquitous, and most versatile family of membrane receptors. This whole thing means that this receptor can bind to many ligands and cause very several effects, okay? And these effects are different than each other. So the G protein coupled receptor can bind to many ligands and cause many effects, okay? And that's what these lines mean. And it's the most common target of therapeutic drugs because it is, it's play roles in many things: cancer, insulin, growth, everything, okay? So it is a common target for many therapeutic drugs because it play roles in many diseases, in many good things, okay? So it's the most common target of therapeutic drugs. Now let's dig, and these are the things that I mentioned. It's 7 transmembrane loops, extra N terminal, binds the ligand, C-terminal, bind the G protein, 3 internal, 3 external, stabilized by disulfide bond, and that's all I mentioned. And let's now, this is the receptor, okay? 7 helicals, N-terminal, C-terminal, disulfide bond, and whatever. We said that at the C-terminal, this is the C-terminal, okay, and this is the 7 things. At the C-terminal, we have G protein coupled to it. What is this G protein? Like, we know the receptor structure. What's the structure of the G protein? And it's complicated, so please focus. G protein is made of 3 subunits. So 3 things make this G protein. First, we have alpha subunit, okay? Then we have beta subunit. Then we have gamma subunit. So G protein is made of 3 subunits, alpha, beta, and gamma. The most important one that cause the action and bind to another things and cause the response is the alpha subunit. So this whole thing is G protein, okay? And it has 3 subunits, alpha, beta, and gamma. The important one is alpha. It causes the action. What inhibits it from causing the action is the beta and gamma. So alpha is actually stimulatory. It go and do the action. Sometimes it can be inhibitory, but most of the time, alpha is causing the action. What inhibits it from causing the action and cause it to be quiet? Beta and gamma, okay? And it's also worth mentioning that this alpha is bind to something called GDB. GDB is guanine disulfide phosphate, okay? Like, you know, ATP, yeah, and ADB, AMB. We have GTB, GDB. And so alpha subunit, it binds to GDB. Okay, so now we have the alpha subunit bind to GDB. When it is bind to the GDB, it's inactive. Okay, so it's inactive because of 2 things. First, because it's bind to the GDB, and because it's bind to the beta and gamma. Once the ligand binds to the N-terminal, it causes this GDB to be converted into GTB by a specific enzyme. We will mention it lastly. So now, once the ligand binds, this GDB gets converted into GTB. And once GTB binds to the alpha, beta and gamma get removed. And now alpha is free and it's bind to GTB. And once it's free and has GTB on it, it go and activate many other enzymes and cause the action. So this is the G protein structure. We have alpha, beta, gamma. Alpha is the stimulatory. Beta and gamma inhibit it from causing the action. Alpha is also bind to GDB and cause it to be inactivated. Once we have the GDB removed and become GTB, and that's when the ligand binds, alpha become free and beta and gamma got removed, and alpha is active and cause many other actions. Is it clear? Okay, that's the G protein structure. Okay, now when it's active, alpha, when it's active, it has GTB with it, and there is no beta and gamma. If we want it to be inactive, like khalas, we do the response, we did everything, we need to finish the response. How? We remove this GTB and put GDB, and once GDB binds to it, beta and gamma binds again, and it becomes inactive and it binds to the C-terminal once again. So when we want to activate it, we remove GDB, put GTB, and beta and gamma got removed, and now the G alpha can go and do the response. Once we want it to be inactive, we remove GTB, put GDB, gamma and beta binds again, and it go and binds to the C-terminal and it's inactivated. So that's the G protein structure. Okay, so here it tells you that first we have the hormone binding to the G protein receptor. Then the G protein complex gets activated by making G alpha free and has GTB with it. Okay, then it goes and activates other enzymes, and these enzymes make another enzymes, and we continue. Okay, and here the slides is just to tell you that the G protein coupled receptors have many ligands can bind to it. Light, calcium, odorants, small molecules, many hormones. So it combines to many things. And based on the thing that it binds, it can cause several responses, okay? And all the responses follow the same pathway. G protein got detached from beta and alpha get detached from beta and gamma, and then it go and cause its action. Okay, so here it says that beta and gamma cause sequestration of the alpha subunit, and it inhibits it from doing the action, and also GDB inhibits it. And once the ligand binds, GDB gets converted to GTB, and alpha gets removed from beta and gamma, and so alpha can go and cause the response. Once we want it to be inactivated, we hydrolyze GTB into GDB, so we convert GTB to GDB, and the beta and gamma binds to the alpha subunit again, and it becomes inactive. Is everything clear? Because now we will talk with specific name of enzymes and things. So I want the whole pathway to be clear in your mind. Is it clear? I want another yes.
Yeah, it's clear.
Okay. So now let's dig deeply into the content. So as I said, we have the membrane, we have the seven helical structure, N-terminal, C-terminal, and the G protein has alpha and beta and gamma into it. Once the ligand binds to the N-terminal, this G alpha has GDB on it. Once the ligand binds, this GDB becomes GTB, and alpha get dissociated from beta and gamma, and so we have G alpha with GTB active. What are the enzymes? GTB. What are the enzymes that remove this GDB and put GTB? We have an enzyme called GEF. Okay, this GEF enzymes remove GDB and put GTB, and so now the G alpha is active and cause the action we want. Okay. Also, this GEF plays role in two ways. It can remove GDB and put GTB, which is activating the G alpha, and it also can inactivate it by removing GTB and put GDB once again, and it become inactive. So GEF enzymes here, it's called the guanine exchange factor. It's just for exchanging. It can exchange the GDB to GTB, so it become active, or it can exchange the GTB to GDB and become inactive. So GEF, guanine exchange factor, play roles in two sides, activating and inhibiting. But most of the time, it is for activating. So GEF activates the G alpha by removing GDB and put GTB. Now, what inhibits it? What makes it inactive? What removes the GTB and put GDB other than the GEF? It's something called GT-base activating protein, or GAP protein here, GAP protein. GAP protein go to the GTB, remove one phosphate, and makes it GDB. So it's inactivated, and we go to the inactive state. So now we have two enzymes, GEF and GAP. GEF can activate and inactivate, but it's mostly for activation by removing GDB and put GTB. The other one is GAP, which is for inactivation. So it removes GTB and it puts GDB. So please memorize these two enzymes because they are very, very, very important. Okay. Now let's talk about the signaling pathway itself. So now, as I said, a ligand binds. Ligands bind to the 7 helical structure. G alpha became free and it has GTB with it. What will it do? It has two main signaling pathways it can go through. There are others, but there are two main pathways. This G alpha GTB, which is active state, either goes to a signaling pathway called adenylyl cyclase pathway, which activates certain enzymes, or it goes and activates another pathway called phospholipase C pathway. So once the ligand bind and G alpha became active with the GTP, it goes to activate the adenylyl cyclase, or it goes to activate the phospholipase C. Okay, so it has two main pathways, adenylyl cyclase and phospholipase C here. Okay, and it does many things, but I just want you now to know the structure of things. Okay, we will explain the signaling pathway in detail afterwards. Okay, so once the ligand binds, G-alpha became active and it activates adenylyl cyclase or phospholipase C, either one of these. And that depends on two things: the ligand that binds and the type of G-alpha. Imagine that this G-alpha has 20 types, but we just want 4, okay? We will talk about them in detail now, but now just focus that G-alpha has many types, and based on the type, it activates either adenylyl cyclase pathway or phospholipase C pathway, okay? And that's it for the second checkpoint. Yeah, before that, the 4 types are GI, GS, GQ, and G12/13. So we have G-alpha S, G-alpha I, G-alpha Q, and G-alpha 12/13. We will talk about them in detail afterward, but now just memorize that we have G-alpha S, G-alpha I, G-alpha Q, and G-alpha 12/13, and each one of them activates certain pathway. Okay, and now to the second checkpoint. So everything clear?
I just have one question.
Yeah, go ahead.
You said GF activates and inactivates the GTP, and GAP just inactivates. When does GAP works and when does GEF works? Or is it just like whenever one of them is there?
Both of them works to inactivate, but GF is not enough because GF is actually busy with activating. Okay, so we need other thing to inactivate, which is GAP. Like GF is not always working in activating, but imagine that we have GAP, like a disease that inhibits GAP. GF now will play a role in inactivating, just to save the day, okay? But as I said, GF mainly is for activating, and GAP is mainly for inactivating. But GAF can inactivate if we needed it to inactivate.
Okay, another question. The adenylyl cyclase and the phospholipase C, what do they exactly do?
We will explain them now after the checkpoint. We will explain the whole signaling pathway with them.
Okay.
But now just know that activate either one of these.
Okay, just one thing. Can we have a praying break? Because I should just.
Do you guys want to pray? I have no problems at all.
I just need to go pray. Is it okay?
Okay, we will have 10 minutes, or do you need more?
I think at 8:15 is good.
Okay, 8:15. Okay, 8:15 we will start again. Can you?
We shut down the Zoom.
Can you stop the recording? Or actually, I can. Just guys, go. No problem. So as was I saying, now we will start talking about the types of G protein. We said that G-alpha can activate one of two pathways, adenylyl cyclase and phospholipase C. It's either one of these. And so G-alpha activates one of these based on the type of G-alpha we have. And so we have 4 types of G-alpha. First, we have G-alpha S, G-alpha I, G-alpha Q, and G-alpha 12/13. And each one here has its type and with its action. So let's talk about them. First, we have G-alpha S, and S here is for stimulatory. Okay, which means that's activating something. This G-alpha, after it get deassociated, it go and stimulate another enzyme. And this G-alpha stimulatory is for adenylyl cyclase pathway. Okay, so G-alpha S, after it get deassociated, it go and activate adenylyl cyclase pathway. This is the first type. Once the ligand binds and we have G-alpha deassociated and went and activate adenylyl cyclase, this means that we have G-alpha S. The other type is G-alpha I, and I here is for inhibitory. Okay, so G-alpha I is for inhibitory, and it goes and inhibits adenylyl cyclase. So once the ligand binds and G-alpha get deassociated and it went and it inhibited adenylyl cyclase, it is G-alpha I, which is G-alpha inhibitory. The fourth type is G-alpha Q, and it goes and it activates phospholipase C pathway. Okay, so the third type is G-alpha Q, and it goes and it activates phospholipase C pathway. And we have something called GT, transducins, and it activates cyclic GMP pathway and phosphodiesterase. And please just memorize that, because actually he said, it's not very important, and you will not take the whole pathway. So we have G-alpha 12/13 or G-alpha T, and both are for cyclic GMP pathway. Okay, so now we have these four types. G-alpha S is for activating adenylyl cyclase. G-alpha I is for inhibiting adenylyl cyclase. GQ is for increasing IB3, which is phospholipase C pathway. And finally, we have GT or G12/13, which is for cyclic GMP or guanylene cyclase pathway. Okay, so now let's dig deeply into each pathway. First, now we will forget about the slide. We will come back later. Now we will take each pathway, and please follow my map. Okay, so now, as I said, we have the membrane. Now we will start with adenylyl cyclase. Okay, so now it's about adenylyl cyclase. And we said that adenylyl cyclase activated by GS. So now we are talking about G-alpha S. So we have the membrane, and we have the 7 helicals or whatever, and it has the N-terminal, C-terminal, and the C-terminals bind the ligand, and the N-terminal bind to the G-alpha, beta, gamma, and GDB. Okay, GDB, yeah. So now the ligand bound to the C-terminal, and GDB got converted into GTP. And now we have G-alpha S deassociated and it go. So now we have G-alpha S activated. Where it go in the adenylyl cyclase? First, G-alpha S go and activate adenylyl cyclase protein. So first it activates adenylyl cyclase. What does adenylyl cyclase do? Now we have adenylyl cyclase that goes to the ATP and converted into cyclic AMP. And cyclic AMP is different than AMP and different than ATP. So first we have G-alpha S go to the adenylyl cyclase and it activates it. Now adenylyl cyclase is an enzyme that goes to ATP and convert it into cyclic AMP. Okay? Now cyclic AMP is a secondary messenger, means that it will go and activate another enzyme. Okay? So secondary messenger here is cyclic AMP, not adenylyl cyclase. Adenylyl cyclase is an enzyme that got activated that formed cyclic AMP. Cyclic AMP now is secondary messenger that will go and activate another enzyme. Cyclic AMP go and activate an enzyme called BKA, okay, or protein kinase A. Protein kinase A. So cyclic AMP go and activate protein kinase A. And from the name, it's a kinase. So it phosphorylates another enzyme. So now PKA, the one that got activated by cyclic AMP, goes and activates another enzyme, and this last enzyme will do the action. And we don't have specific enzyme. We have many enzymes that get activated by PKA. So this is the adenylyl cyclase pathway. So G-alpha S go and activate adenylyl cyclase. Adenylyl cyclase converts ATP into cyclic AMP. Cyclic AMP, as the secondary messenger, go and activate PKA. And PKA phosphorylate a specific enzyme, and that specific enzyme cause the action. This is the adenylyl cyclase pathway. Is it clear? G-alpha S activates adenylyl cyclase. Adenylyl cyclase makes cyclic AMP. Cyclic AMP activate PKA, and PKA phosphorylates the last enzyme, and the last enzyme do its action. Is it clear? I feel it's simple, please. If it's not clear, tell me to repeat it, because that's very important. Okay, so that's the adenylyl cyclase pathway. Now we have a small simple detail that we need to go over, that how cyclic AMP go and activate PKA. Okay, how? Like we know that adenylate cyclase makes cyclic AMP by ATP. It converts ATP into cyclic AMP. Now cyclic AMP go and activate pKa. How? First, let's talk about pKa generally when it's inactive. pKa, or protein kinase A, when it's inactive, it's made of 4 subunits. Okay, so this is the first subunit, this is the second subunit, this is the third subunit, let's say, and this is the fourth subunit. The 2 subunits here are causing the action, and the 2 subunits here are inactivating them. So we have the one that caused the action and the one that's inactivated, bounds to it. Okay, so it's made of 4 subunits, 2 for causing the action, the phosphorylation, and 2 for inactivating them. Now cyclic A and B come and bind here to the things that inactivate. Once cyclic A and B binds to them, it gets deassociated, and now we have the 2 subunits that activate or phosphorylate free. And now they go to the specific protein or enzyme that we need to phosphorylate, and they phosphorylate it. So that's how pKa gets activated by cyclic AMP. Normally, pKa is made of 4 subunits, 2 activatory and 2 inhibitory. The 2 activatory bound to the 2 inhibitory and got inhibited. Now, cyclic AMP binds to the inhibitory. So the inhibitory is gone, and the activatory go and phosphorylate. And once it's phosphorylate, it causes the action. And that's the protein kinase A. So last time I'll repeat it. It's made of 4 subunits, 2 inhibitory, 2 causing the action. The cyclic AMP binds to these inhibitory, and once it deassociates, this activatory go and phosphorylate the enzyme or the thing that we want to phosphorylate, and this enzyme cause the action. Is it clear?
Can you repeat it, please?
Okay. Now, how pKa gets activated? pKa, when it's inactive, it's made of 4 subunits. Let's say this is the first subunit, this is the second subunit, this is the third subunit, and this is the fourth subunit. The 2 subunits that are like a star are inhibitory, and the 2 that are circles are activatory. The activatory is bound to the inhibitory, so they are inhibited. Cyclic A and B come and binds to the inhibitory. Once it binds to them, it causes them to deassociate. And now the activatory are free, and they go and phosphorylate the enzyme that we want to phosphorylate and cause the action. You get me?
Yeah, it's clear now. Thank you.
Okay, so now we need to mention 2 things. Once cyclic A and B binds to the pKa, it becomes active. So how will I inactivate it? We have done the response, everything is now good. We want to inactivate it. We have 2 ways of inactivation. First, we can go to the cyclic AMP and break it down to something called 5-AMP. It's like just a normal AMP. So first, we can break this cyclic AMP and remove it to make pKa go back to the normal state, the inactive state. And how do we break cyclic AMP to 5-AMP? Is by an enzyme called phosphodiesterase. So phosphodiesterase enzyme go and break cyclic AMP to 5-AMP and terminate the action. That's the first way. The other way, we said that pKa go and phosphorylate a specific enzyme, and this specific enzyme causes the action. So if we dephosphorylate this specific enzyme and we remove this phosphate, it's become inactive. And how we dephosphorylate it is by a protein phosphatase. It removes the B, and that's it. So we have 2 ways of terminating the action. First, by breaking down cyclic AMP to 5-AMP, and that's by phosphodiesterase. Once we break it down, pKa is back to normal. The other way is by, we said pKa phosphorylate a specific enzyme. And so when we remove this phosphate group from that specific enzyme, it's become inactive. And how we will do that? By a phosphatase. And that's it. These are the 2 ways that we terminate this action. Is it clear? Okay. Can you please repeat the second one? Okay. So as I said, we have 2 ways to terminate. Since cyclic AMP activates pKa, and pKa go and phosphorylate a specific enzyme, and this enzyme cause the action, we have two ways of terminate this. By breaking cyclic AMP to 5-AMP, and when we break it down by phosphodiesterase enzyme, خلاص, pKa is inactive and خلاص, everything here is gone. This is the first way. The second way, since pKa phosphorylate a specific enzyme to cause the action, if we remove this phosphate group and dephosphorylate, خلاص, there is no action because it's dephosphorylated. And how we do that? By a protein phosphatase. Okay, these are the two ways. So now, I know I'm taking a long time, I guess, but let's recap the last time, the adenylate cyclase pathway. So first, we have the membrane with the G protein coupled receptor. And here's the ligand, and here we have the G protein, which is inactive by GDP and gamma beta. Okay, once the ligand binds, G alpha S gets activated by GEF, and it becomes GTP G alpha S. This go and activate adenylyl cyclase. Adenylyl cyclase go and make cyclic AMP. Cyclic AMP make pKa. And pKa go and phosphorylate a specific enzyme, and that's a specific enzyme. Adenylyl cyclase go to the ATP and make it cyclic AMP, for the one who asked. Okay, and now cyclic AMP make pKa, and pKa phosphorylate a specific enzyme, make it doing its action. Okay, so now this is the adenylyl cyclase. We have 4 ways of terminating it. First 2 ways we mentioned them before the break, which are the GEF that can revert GTP to GDP, and the GAP protein, if you remember. It reverts GTP to GDP, and that's terminating the whole thing. Or we have the phosphodiesterase, which breaks cyclic AMP to 5-AMP, and we have phosphatase, which remove the phosphate group. And these are the 4 ways of terminating. Okay, and that's the adenylyl cyclase pathway. Is it clear? Because now we will start the other pathway. So please, first pathway must be clear. Is it clear? Okay. Let's continue. So that's the first pathway. Now we have another pathway in adenylyl cyclase, which is GIS. Sorry, not GIS, G alpha I. Yes, it's G alpha I. G alpha I is inhibitory. Okay, so it inhibits adenylyl cyclase, so we don't have any other thing. خلاص, that's it. So we have G alpha I that go and inhibit adenylyl cyclase, and خلاص, it inhibits the whole thing. So this is the 5th way of inhibiting. G alpha I go inhibit adenylyl cyclase, and خلاص, we don't have any action anymore. Okay, and that's G alpha I. Now we will talk about phospholipase C pathway. So first, as I said, we have the membrane, we have the 7 helical, we have the ligand here bind, and here we have G alpha Q. Okay, so when the ligand bind, G alpha Q gets activated, and now we have an activated G alpha Q with GTP. G alpha Q, we said it's for phospholipase C pathway. What does happen in this pathway? So in this pathway here, this is the, where is it? Yeah, this is the picture of it. Okay, so in this pathway, G alpha Q go and activate phospholipase C protein. Okay, so it activates a protein called phospholipase C. Phospholipase C protein go and break something called PEP2. Okay, it go to PEP2, and since it's 2, once we break it, it will give us 2 different things. First thing is DAG. Okay, DAG. The other thing is IB3, inositol. We call it inositol triphosphate. Okay, it's IB3 or inositol, and DAG. So phospholipase C got activated by G alpha Q. Phospholipase C go and break PEP2 into IB3 and DAG. Now, what does happen now? DAG go into the membrane and stay there. Okay, and IB3, which is inositol, go, and as you took in MSK, calcium is inside the cell under in something called sarcoplasmic reticulum or something like that. So IB3 go and open the channel and cause calcium to get out. Oh, okay. And now DAG and calcium together activate something called PKC, phospholipase, protein kinase C. It activates protein kinase C. And protein kinase C phosphorylates another enzyme, and that enzyme causes the action. So let's recap. G alpha Q activate phospholipase C. Phospholipase C break PEP2, and PEP2 break down to IB3 and DAG. DAG go to the membrane and stay there. IB3 go inside and cause calcium to be released from sarcoplasmic reticulum, and both calcium and DAG work together and activate protein kinase C, and protein kinase C phosphorylate and cause the action. Is it clear? It's a complicated one, but I hope it's clear. It's clear. Okay. Okay. Now, there in the adenylate cyclase, we said that cyclic A and B is a secondary messenger. What is the secondary messenger here? We have IB3 as secondary messenger because it goes and causes calcium to be released. We have DAG as secondary messenger, and we have calcium itself as a secondary messenger. Calcium and DAG as secondary messenger activate PKC, and PKC phosphorylates an enzyme and cause the action. And so this is the whole diagram that summarized the whole thing. And here, as we said, calcium is a secondary messenger. Normally, inside the cell, there is very low calcium. And outside the cell, we have very high calcium. But inside the cell, we have sarcoplasmic reticulum. And once inositol go here, it causes calcium to be released from inside the cell into the cytoplasm. And now calcium is very high. And with the work of DAG, it activates PKC. And PKC phosphorylates an enzyme and cause the action. And that's phospholipase C whole pathway. Is it clear? Because now we reached the third checkpoint. I guess we have one more checkpoint, and that's it. So, and that's it for this lecture, by the way. We have 2 other lectures. So is it clear? Okay, I want another yes. Okay, so that's it for these 2 pathways, adenylate cyclase and phospholipase C. We said we have another pathway, which is G12/13 or GT, but we will explain it at the end of the lecture because then it's a little bit irrelevant. So now problems with signaling. We know Ahmed Jada doesn't like just the whole pathway. He loves to put some clinical and diseases and these things. So now we have some diseases in this pathway. You can't know what are the diseases doing except if you knew. Wait, we have a question here. IB3, DAG, and calcium are all second messengers. Yes, IB3, DAG, and calcium are all second messengers. Second messenger means that it goes, move all the way, and activate another thing. So IB3 go and activate the channels that cause calcium to be released. Calcium and DAG go together and activate PKC. Okay, so all are second messenger, yes. So, yeah, welcome. So now, if we know the pathway, we must know the diseases. The diseases are happening due to 2 things. Either we can't inhibit, so the enzymes are always working. And since the enzymes are always working, it causes many problems, okay? Or the enzymes are not working and we don't have an action. So we have 2 types of diseases. First, the increase in activity. So we have a lot of activity, and that's due to 2 things. Either that's excess on signal, like we have a lot of activating signal, which means that we have a lot of activation, or lack of signal. So we can't inhibit. So everything is still continuing till the very end. So we have these 2, increasing activity, because when you have increase on signal, you have very high activity. And if you can't make an off signal and inhibit this activity, it will remain high. So this is the first type of disease, increased activity. The second type, which is we don't have any examples here in the lecture, is the decreased activity or absence. So the G protein is not active or the protein kinase is not active. So we don't have any activity, so we don't have any response. So the ligand is not binding, the receptor have a problem, the G protein is not working, so it is decreased activity. So these are the two types of disease. We have two examples, very important, cholera and pertussis. And both of them are due to increase in activity. Okay, so we have cholera and pertussis, which is whooping cough. And these two diseases are due to lack of disease. We can't stop. Okay, so everything is continuing till the very end. So lack of an off-stop signal causing cholera and pertussis. So let's get into each one. First, as I said, we have two ways. We have actually four with adenylate cyclase, but mainly for the G protein, we have two ways of inhibiting G protein. First, hydrolysis of GTP to GDP, and that's by GEF or GAP, mainly GAP. And so when we convert GTP to GDP, everything is inhibited, okay, or inactive, yani. Or the other way is by activating inhibitory G protein, which is GI, okay, G alpha I. We have another ways, but now these diseases depends on these two ways, okay. So we have two ways. First, hydrolysis of GTP to GDP, so everything is inactive after that, or activating of G alpha I, which inactivate everything, okay. So let's start with cholera. So cholera is a bacteria, okay? It's released toxin. This toxin and it inhibits the hydrolysis of GTP to GDP. So now we can't convert GTP back to GDP. So GTP will stay there till like forever, and it will cause the activation of cyclic AMP all the time, okay? And since cyclic AMP is not inhibited and it's activated all the time, it will go and activate a receptor called CFTR, and CFTR will cause chloride to get out and water to get out with it. And because chloride and water are getting out, it causes diarrhea, okay? It causes efflux of ion and water and cause diarrhea. So that's cholera. Cholera release a toxin. This toxin inhibits the conversion of GTP to GDP. Once it inhibits that, the cyclic AMP will remain high till like forever. And cyclic AMP will go to the CFTR and open them and cause efflux of chloride and water and cause diarrhea. This is cholera. Is it clear? Is cholera clear? Because he will bring a question about it.
Yeah, it's clear.
Okay, so the other thing is pertussis. Pertussis release a toxin. This toxin inhibits the activation of G alpha I. G alpha I, we said it goes and inhibits adenylate cyclase. So once pertussis toxin inhibits G alpha I, adenylate cyclase will become active all the time. And so we have cyclic AMP all the time. And because of that, we have overproduction of cyclic AMP, lead to several systemic effects. Among of them is the increase in insulin and hypoglycemia and pertussis, which is whooping cough. So pertussis release a toxin. This toxin inhibits G alpha I, and the inhibition of G alpha I will cause adenylate cyclase to be active all the time, which cause pertussis, whooping cough, increase in insulin, and hypoglycemia, which is low glucose because of high insulin. Is it clear?
Pertussis isn't.
What?
Pertussis isn't clear.
Okay,
pertussis.
Let's recap both of them. Cholera release a toxin. Okay, this toxin that cholera release, it inhibits GDP conversion to GTP. Once it's inhibited, it inhibits the conversion of GTP to GDP. Once that's inhibited, the GTP will remain, and the cyclic AMP will get overstimulated all the time and open CFTR receptors. And once they are open, chloride and water will get efflux, and we will have diarrhea. This is one. This is cholera. Pertussis release a toxin. This toxin inhibits G alpha I. G-alpha I inhibits adenylate cyclase. So if we inhibit it, we call it inhibitory of the inhibitor. Isn't G-alpha I an inhibitor? If we inhibited the inhibitor, we will have overstimulation. So when we inhibit the inhibitor, which is G-alpha I, we will have overstimulation, and this overstimulation will cause pertussis and high insulin and hypoglycemia, but mainly pertussis. Is it clear?
Yeah, thank you.
Okay. So now we reach to the very last checkpoint. We have one more thing, and that's it for the first lecture. So yalla, let's continue. We have something called NO, nitric oxide. You took it a lot of times, especially in MSK, you have nitric oxide all of the time. Yeah. Let's talk mainly about nitric oxide before we talk about what's the pathway that it causes. Nitric oxide causes vascular reactions. Actually, it goes and causes vasodilation. That's what you took all of the time. So nitric oxide causes vascular actions, including direct vasodilation. It goes and binds and causes a specific pathway we will talk about. And that pathway ends up by vasodilation. Okay? The other thing is indirect also vasodilation, and that's by inhibiting vasoconstriction. So nitric oxide causes vasodilation, and it inhibits vasoconstriction, which also causes vasodilation. Okay? And also it has antithrombotic effects, which inhibit platelets and thrombosis, these things you took in HLS. And it has anti-inflammatory effect. So it inhibits inflammation, okay? Inhibits leukocyte adhesion and vascular endothelium, and these things that are included in inflammation. And anti-proliferative effect, which inhibits hyperplasia, inhibits like the growth of the muscles. So nitric oxide have 5 effects. He can ask you about them in SAQ. So it causes direct vasodilation, which vasodilate the vessels, indirect vasodilation by inhibiting vasoconstriction, antithrombotic effect, which inhibits platelets adhesion, anti-inflammatory, which inhibits inflammation, and anti-proliferative, which inhibits muscle hyperplasia. Okay, these are the 5 effects of nitric oxide. Now, nitric oxide, we have it in our body. How? We are synthesizing it from something called nitroglycine. Okay, so in our body, we have nitroglycine. We convert it into nitric oxide, and that nitric oxide make these 5 actions. We can take nitroglycine in condition like cardiovascular ischemia, and it acts by making nitric oxide. Like we can take nitroglycine as a medicine, okay, or as a drug, but it's not very effective, okay? What we can take other than that is aspirin. Why? Aspirin is an NSAID that causes NO production. So we can take nitroglycine, and in our body, it gets affected to nitric oxide and cause these 5 effects. Or we can take aspirin, and aspirin is an NSAID that causes nitric oxide production. And once nitric oxide is produced, we have these 5 effects. So now we know what nitric oxide do. How does it do these effects? So simple. Just a sec. I told you a little bit about something called G-alpha 12 over 13, or G-alpha T. We took adenylyl cyclase in GS. Here we have something similar to adenylyl cyclase, which is called guanylyl cyclase. You know adenosine, guanine. Okay, here we have adenylyl cyclase and guanylyl cyclase. Okay, so G-alpha T or G-alpha 12 over 13 cause the same reaction of GS, but not adenylyl cyclase, but guanylyl cyclase. So how does nitric oxide do its work? Nitric oxide is the ligand that go and bind to the G protein coupled receptor, the 7-alpha helical thing. It causes the association of G-alpha T or G-alpha 12 over 13. Now, G-alpha T or G-alpha 12 over 13 go and doesn't activate adenylyl cyclase, it activates guanylyl cyclase. Okay? Guanylyl cyclase. Now, guanylyl cyclase doesn't do cyclic A and B, it do cyclic G and B. Because that was adenylyl, so it's cyclic A and B. Now we have guanylyl cyclase. It makes cyclic GMB. Now cyclic GMB go all the way and activate something called protein kinase G, okay? And protein kinase G cause the phosphorylation and this thing. So adenylyl cyclase activates cyclic A and B, and cyclic A and B make pKa. Guanylyl cyclase activates cyclic G and B, and cyclic G and B activates pKg. Is it clear? Just remove every A and put G. That's it. And that's how nitric oxide work. It go activate G alpha T and G alpha 12 over 13. They activate guanylyl cyclase. Guanylyl cyclase activates cyclic GMB, and cyclic GMB activates pKg. And pKg phosphorylates other enzymes, and that's it. Okay. And it's also worth mentioning that since cyclic A and B is made of ATP, we convert ATP to cyclic A and B. So cyclic GMB is made of GTP. Okay, so everything is converted to a G. Adenylyl, guanylyl, cyclic A and B, cyclic G and B, pKa, pKg, and that's it. For the NO, nitric oxide. Is it clear? Okay, now we have something I hate, personally I hate, and it's kinda a little bit hard, so please pay attention because it's mixed of many receptors. We took that we have the 7 helical, we took that G protein coupled receptor is 7 helical structure, and it has a C terminal and an N terminal. C terminal is for the ligand, and N terminal is for G protein. I told you that G protein made of 3 things: alpha, beta, gamma. Gamma. Yeah, alpha, beta, and gamma, which is trimeric. What trimeric mean? Trimeric mean that it's made of 3 structures: alpha, beta, gamma. So G protein is trimeric, made of alpha, beta, and gamma. We have another G protein, which is also bind to G protein coupled receptor, but it's not trimeric. No, it is monomeric. Monomeric means that it's made of 1 subunit, and that subunit is Ras. Not alpha, not beta, not gamma. Something called Ras. Ras G protein. Okay? So we have 2 types of G protein. The one we took all the time, which is adenylate cyclase, phospholipase C, and these things, which is the trimeric, made of 3 subunits, alpha, beta, gamma. And the another one is the monomeric, and it's made of 1 molecule or 1 subunit, which is Ras. And this is it, the Ras G protein. And it has complicated, let's say, pathway. So let's take it now. I guess it's the last thing in this first lecture. The other lecture is way much easier than this, so let's focus. We said that we have 4 types of receptors. First one is the ion-gated channels. It's an ion bind to the channel, open it, and cause calcium, Na, or whatever to enter and get out. We said that we have G protein coupled receptors. G protein coupled receptors is an alpha helic or 7 alpha helic or whatever, and it has G protein with it. We took that we have the steroids, but my friends Amjad will explain them. And we took that we have enzymatic receptors, or actually tyrosine kinase receptor. Tyrosine kinase receptor have a special thing that this is the receptor. Okay, this is the receptor, and it has a tyrosine kinase enzyme inside it. Once the ligand binds, the tyrosine kinase phosphorylates the enzymes inside and cause the action. So once the ligand binds, the tyrosine kinase gets activated and it phosphorylates many other enzymes. Okay, so the Ras, this one, the Ras, the G protein Ras, is a mixture between G protein and tyrosine kinase. How? I will tell you how. We have the membrane and we have the G protein or whatever. Okay, and we have Ras here. Bind, it's not bind actually. G protein Ras can't bind. We have the receptor. It's like this. Once the ligand bind, the tyrosine kinase gets activated. Tyrosine kinase gets activated. Hello? And when it's activated, it phosphorylates. It must phosphorylate Ras. Okay? But there is one rule here. To be phosphorylated by the tyrosine kinase, you must have something that makes you bind to the tyrosine kinase. And that thing is something called SH2. We will talk it more in the next lecture, but now just know that any molecule must have SH2 as a domain of, this is the whole molecule, and some part of it called SH2. This SH2 can bind to the tyrosine kinase and cause phosphorylation of this whole thing. To be phosphorylated by tyrosine kinase, you must have SH2. The issue that Ras doesn't have SH2, okay? So it can't bind. So it must use something called adapter protein. Adapter protein is something that has SH2, go and be phosphorylated, and then go to the Ras and phosphorylate Ras. And that's the GRB2. Okay, I know it's very complicated, but we will keep repeating it till you understand. So we have the receptor. This is the receptor. Okay, once the ligand binds, tyrosine kinase gets activated. Tyrosine kinase wants to phosphorylate, and so it must phosphorylate Ras. But the issue that Ras doesn't have something called SH2, which can bind to the tyrosine kinase and cause phosphorylation of Ras. So how can Ras be phosphorylated? Is by using an adapter protein. An adapter protein means that it gets phosphorylated, then goes again to the Ras and phosphorylates Ras. And this adapter protein in Ras pathway is GRP2. GRP2 gets phosphorylated because it has the SH2. And once it gets phosphorylated, it goes to the Ras and phosphorylates Ras. And once Ras is phosphorylated, it goes and activates enzymes, which is RAF and MABIC-ERK. He said he will not ask you about them, but if I were you, I will just memorize them. So Ras, once it's activated, it binds to the GTP. It's activated, it phosphorylates RAF and MABIC-ERK, and that's it. That's the pathway. MABIC-ERK go and cause its action. So last time, I will say the pathway because we took a long time here. We have the receptor. The ligand binds to it. It activates tyrosine kinase inside. Tyrosine kinase wants to phosphorylate Ras, but Ras doesn't have SH2. So it uses an adapter protein that has SH2, which is GRB2. GRB2 go to tyrosine kinase, be phosphorylated, and then go to Ras and phosphorylate Ras. Ras, it's similar, it's a G protein. G protein, when it binds GDB, it's inactive. When we phosphorylate it to GTP, it becomes active. Same thing with Ras. When we phosphorylate Ras, it becomes GTP, and it go and activate RAF and MABIC-ERK. Okay, and RAF and MABIC-ERK cause the whole signaling pathway, and that's it. RAF activates MABIC-ERK, MABIC-ERK cause gene activation and these things, and that's it. To inactivate it, we hydrolyze GTP to GDP, and we inphosphorylate RAF, not RAF, we inphosphorylate Ras, and that's it. Why Ras is important? Because Ras, this one Ras, the G protein Ras, is activated in tumors. When Ras is very active or overactive because we can't inhibit it, it causes tumors or malignancy. So now we have three diseases: cholera, pertussis, and tumors. Cholera, we can't inactivate the G protein, the normal G protein. Same with pertussis. But tumors is because we can't inactivate the Ras. Okay, so Ras is overactive and it go and cause malignancy. Is it clear? Okay. So now we finished the first lecture. We have question here, but I will go on them once we finish the whole thing because this question is for both lectures. So do you want a break of 5 minutes? Like we start on 9:06 or 9:05 and we start the second lecture. Okay, you want that? Okay, we will start at actually 9:07. So see you soon. So now we finished the first lecture. So now we will start with the other lecture. In the first lecture, we talked about two types of enzymes, the ion-gated channels a little bit, as an ion binds or a ligand binds, open, and ion enter and get out. And we talk deeply about G protein coupled receptor and a little bit about catalytic receptor, which are the tyrosine kinase, because we wanted to explain the Ras. Now we will talk deeply about the tyrosine kinase receptors, so pay attention. So at the beginning of the lecture, they just give an overview that we have membranous receptor and it binds the ligand and it causes internal signaling pathway of many enzymes and second messengers. So it causes amplification. At the end, it causes a response. And that we have these types of receptors. So we explained ion channel, G protein. Now we have enzymes. So this lecture is about one type of receptor. So it's kind of lighter lecture. So we will finish fast, inshallah. And we have others, which are the steroids, and my friend Amjad will explain them. So this is the last recap before we start. First, we have ion channels. So it binds an ion. Then once it binds the, it binds the ligand. Once it binds the ligand, which is the red thing here, once it binds the ligand, it opens and the ions enter. The other type is the G protein coupled receptor. So it has G protein coupled to it. And once the ligand binds, the G protein move and cause the action. The last type we have is the enzyme-linked receptor. And now we are starting the new thing. I told you a little bit about it to explain the Ras, but we didn't talk about the structure or anything. So let's now talk deeply about it. So please pay attention and let's start. We have for the tyrosine kinase receptor, or mainly the enzymatic receptor. The enzymatic receptor is special because they have the whole receptor itself has enzymes in it at the end of it. So these enzymes are usually kinases that phosphorylate many other enzymes. So once the ligand bind, these kinases get activated and it causes phosphorylation. What is the structure? The structure is like this. We have the membrane, and this membrane has two different receptors. This is the first receptor, and this is the second receptor. These receptors are not attached to each other, and each one of them has tyrosine kinase at the end of it. So it has the tyrosine kinase at the end of it that causes phosphorylation. Once a ligand binds, the ligand must be a dimer, okay? So it can't be like just one ligand. It must be a dimer, so it has two molecules. Once this dimers bind, it binds to both of the receptors together, and it causes dimerization, means that the two receptors that are not attached to each other will attach. So now after the binding of this dimer, it causes dimerization and the receptor become connected to each other. The two receptors get attached to each other. And once they got attached to each other, each one of them has a tyrosine kinase, right? It causes something called autophosphorylation. Autophosphorylation means that this tyrosine kinase on the left side phosphorylates the right side. And the tyrosine kinase on the right side phosphorylates the receptor on the left side. So each one of them like shake hands with each other. So the first one phosphorylates the other one, and the other one phosphorylates the first one. And that's the autophosphorylation. So the tyrosine kinase receptor or enzymatic receptors must bind a dimer. Once it binds the dimer, which has two molecules, it gets dimerized. So two receptors get dimerized together and attached to each other. And each one of them phosphorylate the other one, causing phosphorylation. So it shakes hands with each other. The first tyrosine kinase autophosphorylate the second one, and the second one phosphorylate the first one, and it become activated. It's worth mentioning that, the enzymes is a protein, and the protein is made of a whole sequence of amino acids. So this tyrosine kinase, this enzymatic receptor is made of many amino acids. One of these amino acids, it's tyrosine. And when I say tyrosine kinase, means that at the tyrosine amino acid of this receptor, there is a kinase. And this kinase phosphorylates other proteins and other receptors. So as I said, we have the membrane and we have the receptor. The receptor has a tyrosine kinase receptor, which means that at the tyrosine residue, we have a kinase. This kinase phosphorylates other things once the ligands bind here. Okay? We may have serine kinase or glutamate kinase, which means at the glutamate, we have a kinase. At the serine, we have a kinase, but the most common one and the most effective one is the tyrosine kinase, which is a kinase on the tyrosine residue. Is it clear? The whole structure or whatever. Okay, so that's the basic structure. Let's now dig deeply into it. Okay, what is this? Okay, so now we have the membrane. The tyrosine kinase receptor or enzymatic receptor made of three parts. The first part is the N-terminal, which is this, N-terminal, which is outside, okay, the membrane. Then we have the transmembrane part, and the transmembrane is usually helical. And we have the intracellular part, and the intracellular part has the tyrosine kinase. So it has three parts, the N-terminal, which is the binding site. We have the alpha helical, which is the transmembrane part, and we have the intracellular part, which has the C-terminal and has the tyrosine kinase. And when the ligand binds to the N-terminal, it must be a dimer. So it binds on both sides. When it binds on both sides, the two receptors attach to each other and cause autophosphorylation and phosphorylate each other. So it means of three parts, N-terminal, transmembrane, and the C-terminal, which has the tyrosine kinase. Okay, and this whole thing is said here. Now we said that this receptor, it's a receptor. This receptor has the tyrosine kinase. So this receptor itself causes phosphorylation of another enzyme. So it is two things, a receptor and an enzyme, which is a kinase. So it's a receptor that binds things, and also it is a kinase that phosphorylates things. We have other things called non-receptor tyrosine kinase. So it's not a receptor, but it is a tyrosine kinase. So it is things that has a tyrosine kinase, which phosphorylate other things, but it's not a receptor. It is just a molecule that swim in the cytosol. And that's like the GRB2, the one we explained in the Ras. It's not a receptor, but it goes here, get phosphorylated, and then by its tyrosine kinase, it go and phosphorylate other things. So we have two types of tyrosine kinase. Receptor tyrosine kinase, which is a receptor that has a tyrosine kinase, and we have non-receptor tyrosine kinase, which is not a receptor, but it's a molecule that has tyrosine kinase, and this molecule, when it gets activated, it go and phosphorylate other things. Okay, and that is said here. No, actually, it's not said here. It's said after two slides. Okay, so is it clear? Okay, so here, here we have that the receptor can have tyrosine kinase, but it can have other enzymes, not just tyrosine kinase. As I said, it may have serine kinase, threonine kinase, and many other proteins. But the main one is tyrosine kinase. So as I said, the receptor has an N-terminal, which is outside, transmembrane helical, and a C-terminal that has tyrosine kinase in it. Once the ligand bind, these autophosphorylate each other and become active. So these are the three parts. I talked about them. This slide is just to tell you that no matter what the enzyme is inside, it's tyrosine kinase, it is kinase something else, serine kinase, no matter what the enzyme is, it has the same structure, a ligand side outside, a transmembrane alpha helical, and a C-terminal inside. But most likely, it will have tyrosine kinase domain, most likely, okay? So this is the first checkpoint. We still have just two checkpoints. Actually, we finished half of the lecture. So is it clear? Okay. So now let's talk deeply into the mechanism of action. I said in the Ras lecture that tyrosine kinase, we said that we have the membrane and whatever, this, just a second. And here we have the tyrosine kinase. The aim of this tyrosine kinase is to phosphorylate other enzymes. These other enzymes that will get phosphorylated by this tyrosine kinase must have something called SH2. If they doesn't have SH2, they can't bind here and they can't be phosphorylated. They must use something called adapter protein. So we have two types of things that get phosphorylated by tyrosine kinase. First, we have the things that have SH2. They go and they get phosphorylated, and that's it. When they get phosphorylated, they go and cause their action. We have things that they doesn't have SH2. And since they doesn't have SH2, like Ras, they can't go and get phosphorylated. So what they use wasta or faza. What is this wasta or faza? Is the adapter protein. Adapter protein, like GPR2, there are many adapter proteins. This is just one of them. It has CH2. It go and get phosphorylated by tyrosine kinase, and then it goes to Ras and phosphorylate Ras. It take this P and send it to Ras. This is the wasta or faza that Ras use, is the GBR2. So we have two things. Things that have SH2, they go bind, get phosphorylated, and that's it. And things that doesn't have SH2, like Ras, they use adapter proteins, and these adapter proteins must have SH2. They get phosphorylated and then give it to Ras, and that's it. Okay, so here we said when it's phosphorylated, we have tyrosine kinase. Tyrosine kinase phosphorylate the things that have SH2, whether it's a normal thing or the adapter protein. If it's the normal thing, once it's phosphorylated, and go and cause the action. If it is an adapter protein, it take this P and give it to Ras or whatever thing that need the adapter protein. Okay. Now we have something called non-receptor tyrosine kinase. I explained it, but let's reinforce in it. We have receptor tyrosine kinase, which is a receptor that has tyrosine kinase, which phosphorylate thing, but it's also a receptor. We have non-receptor tyrosine kinase, which are things that has tyrosine kinase, but they are not receptors. They are just a molecule here. So a molecule that has tyrosine kinase. Okay, it's like SH2. SH2 is a non-receptor tyrosine kinase. Anything that has SH2 and it's an adapter protein is a non-receptor tyrosine kinase. It get phosphorylated here, then it use its tyrosine kinase to phosphorylate Ras or whatever other thing. So we have SH2, which is a non-receptor tyrosine kinase, something that it's not a receptor, but it is a tyrosine kinase because it has tyrosine kinase. So once it's activated, it uses its tyrosine kinase to activate other things. This SH2 is special. Why? Because it's made of three things. Since it's SH2, we surely have SH1. So it's like this. It's SH1 bind to connected to SH2, connected to SH3. SH2 and SH3 both can bind to tyrosine kinase receptor, and both have tyrosine kinase, and they cause phosphorylation. So it's like here. We have SH2, SH3, and SH1. We said SH2 is necessary to bind to the receptor tyrosine kinase. Also, SH3 can bind, and SH1 is the tyrosine kinase that causes the phosphorylation. Okay? Other things other than SH2 that are non-receptor tyrosine kinase, which mean that they are tyrosine kinase and they can bind to the receptor tyrosine kinase, are genus kinase family, which is JAKs. Okay? So SRC, which are the SH2, and JAKs are non-receptor tyrosine kinase, means that they are tyrosine kinase, but they are not a receptor. They go and bind to the tyrosine kinase receptor, become phosphorylated, and then they use their own tyrosine kinase to phosphorylate another thing. So they are adapter proteins. Is it clear? So I feel like I messed around a little here. I took a long time. So is it clear? Okay, repeat last part. So we have two things. We said that we have a receptor tyrosine kinase, which means that it's a receptor that has tyrosine kinase in it, which phosphorylates. And we have non-receptor tyrosine kinase, means that it's not a receptor, but it has tyrosine kinase, which means that it can phosphorylate, like SH2, like GRB2, which has SH2 in it, and JAKs. These things are not a receptor, but they have tyrosine kinase, which means that they can use it to phosphorylate other things. How do they function? They go and bind here. to the receptor tyrosine kinase. When they get phosphorylated, they use their own tyrosine kinase and phosphorylate other things. So they act like adapter proteins. Okay, is it clear? Okay. So let's talk about important adapter molecules. We already talked about them, which are CH2 and CH3. We said CH2 is important to be able to bind into tyrosine kinase. So anything that binds to tyrosine kinase must have SH2, which is a normal thing or an adapter protein. It must have CH2 or CH3. CH3 is bound to CH2, so both work together. But mainly the one important to be able to bind is CH2. So if they ask you in MCQ, what is the thing that exists in adapter protein that allow them to bind to tyrosine kinase receptor? It's SH2, not SH3. Say SH2, it's more accurate. And SH2 is for SRC homology domain. Okay. Other than SH2, we have the Jak-Stat. Okay, or Jak. We already mentioned Jak as an adapter protein, and it needs to bind, then it has its own tyrosine kinase to activate. This line is not so correct. Here two important things that needs adapter molecules, which are Ras and Stat. We said Ras doesn't have SH2, so it uses an adapter protein, which is GRB2. Okay. Other than that, we have Stat that use Jak, okay, as an adapter protein. So this line is wrong. It's two important things that need adapter molecules, which are Ras, that needs GRB2, and Stat, which need Jak. Okay. Now we have the whole pathway of Ras and Jak again. We already explained Ras, so I will just go over it fastly. We said that we have the membrane and we have the receptor tyrosine kinase. It's like this. The molecule bind and it became autophosphorylated, and we have tyrosine kinase. Tyrosine kinase aim to phosphorylate Ras, but the issue with Ras that it doesn't have SH2. So how can Ras be phosphorylated by tyrosine kinase while it doesn't have SH2? It uses an adapter protein called GBR2. GBR2 go and it has SH2, bind to the tyrosine kinase, became phosphorylated, and then with its own phosphorylation, go and phosphorylate Ras. Now Ras, when it's phosphorylated, Ras itself go and activate RAF, and RAF activate Mabek-ERK pathway. And these things need to be memorized, okay? Although he said he will not ask about Mabek-ERK and RAF, but just memorize that Ras activate RAF, and RAF activate Mabek-ERK, and Mabek-ERK go and cause the gene whatever reaction. Okay, he didn't talk about SOS, so I will not SOS. He didn't talk about it, so I will not say anything. But SOS work with GRB2. Okay, so GRB2 before it binds to RAS, it binds to its SOS, but it's not important. Now let's talk about the Jak-Stat pathway, and it's very important pathway. Okay, and it's easy. Okay, let's start. So we have the membrane, we have the tyrosine kinase, we have the ligand binding, and the tyrosine kinase gets activated. The aim of tyrosine kinase is to activate Stat. Okay, just a second here. I want to make sure of one thing. Yeah. Okay. And it wants to activate Stat. Luckily enough, Stat has SH2. So that's good. It's not Ras. Ras doesn't have SH2, so it needs an adapter protein. But Stat has SH2. So SH2 can go and bind here and be activated with Stat. So Stat can bind to the receptor tyrosine kinase and be activated. Good. Then why do we need Jak? Stat need a lot of phosphorylation. So Jak go and bind here to the receptor tyrosine kinase, and it didn't get phosphorylated. Actually, Jak go and it gets phosphorylated, sure, but once it gets phosphorylated, it phosphorylate tyrosine kinase back, okay, to amplificate the response, okay? So now tyrosine kinase is more active, and it phosphorylates Stat by its SH2, and then this Stat bind to another Stat on the SH2, so it's like this Stat, and it's SH2 connected to another SH2 with its Stat. And now this whole thing work to activate enzymes and these things. And this is summarized here. We have this receptor and the tyrosine kinase got activated. Jak come and bind. Once Jak bind, it phosphorylates and it gets phosphorylated back. So it's like a cycle to increase the phosphorylation. Then once everything has a very high phosphorylation, it phosphorylates stat by its SH2, and then stat bind with another stat, and together they cause the reaction. Is it clear? And this is the Jak-Stat pathway, the whole thing. Here it says Jak phosphorylates and activates stat. So Jak is actually doing many things. Jak go to the receptor, activates it, and phosphorylates it more. And also Jak can go to the SH2 and also phosphorylate it, because we said Jak is an adapter protein. So Jak can do two things. Jak can go to the receptor tyrosine kinase, phosphorylate it and activate it more, yani. And Jak also can go to stat and phosphorylate it. And stat can be phosphorylated by tyrosine kinase alone. But to increase the phosphorylation, Jak also helps. So Jak is the ultimate adapter protein. It can do two things: phosphorylate the tyrosine kinase and phosphorylate stat. Is it clear? This is very important pathway because they love to ask about Jak-Stat. Yes. Okay, and this is the whole thing summarized as the Jak-Stat pathway. Now we just have two pathways left. I know it's many pathways, but once you memorize them, everything is good. Once you understand, actually, everything is good. This pathway is easy, very simple. So let's take it. It's called the BI3 pathway. When we took this name, we didn't take this name, actually. It's something similar to it called IB3 inositol. We took it in the phospholipase C. So please don't confuse it. IB3 inositol is different than BI3. They have similar names, but they are way much different, okay? So BI3 pathway is a way much different pathway, and it's related to a tyrosine kinase. So how does it work? It's like this. We have the membrane, we have the tyrosine kinase receptor, and once the ligand binds, the tyrosine kinase gets activated. Once the tyrosine kinase activated, it phosphorylates something called BI3. Okay, it's actually BI3K. Okay, BI3K. It's here the wrong is wrong. I remember that I said it's wrong. It's BI3K. Because it's a kinase. It's BI3K. So please don't confuse it with IB3. IB3 is just something, but BI3K is a kinase. So once this kinase is phosphorylated by tyrosine kinase, it can go and phosphorylate other things. So it goes to something called BEP2. We took BEP2 in phospholipase C. Phospholipase C go to BEP2 and break it down to two things. But now BI3K is a kinase. It can't break it down. Since BI3K is a kinase, it go here and make it BIB3. Because it's a kinase, it's add a B. So now we have BIB3. So again, ligand bind, tyrosine kinase is activated, tyrosine kinase activates BI3K, BI3K go to PEP2 and phosphorylated to making it PEP3. Okay, now this PEP3 can do one important thing. This PEP3 go and phosphorylate or activate something called ACT. Okay, and this ACT can go and phosphorylate one last thing. This one is bad. Okay, so PEP3, it's a reversed arrow. PEP3 can go and activate ACT, is recruited by PEP3. See, the wrong is wrong. PEP3 can go and phosphorylate ACT, and ACT activate bad, and bad do the following actions. Okay, and one last thing to mention, BTEN can go to PEP3 and convert it back to PEP2 to inhibit the whole thing. How does it convert it back? By a phosphatase action, which dephosphorylation. So BTEN is a phosphatase that converts PEP3 to PEP2 to end the reaction. So this is the last time I will mention this before the checkpoint. The whole BI3K pathway is like this, because we have one last pathway, which is the continuation of BI3K. So please focus. We have the membrane, we have the tyrosine kinase, and the tyrosine kinase, the ligand bind, autophosphorylation happen, and we have tyrosine kinase here. Tyrosine kinase phosphorylates PI3K. PI3K is a kinase. Go and phosphorylate PEP2 into PEP3. Now PEP3 go and activate ACT, and ACT phosphorylates bad, or activates bad, and bad do the following action. Then if we want to end this, we use BTEN. BTEN is a phosphatase that converts PEP3 to PEP2 back, and it ends the whole thing. Okay, is it clear? It's a simple pathway, it's because it's after many pathways, it becomes a little bit complicated. Okay, so everything is clear. So now we reach to the last checkpoint, then we have the insulin pathway, and that's it for my session. So do you have any questions? Anything you want me to redo? Okay. So now to the, just a second. Okay. So now to the last thing, which is insulin signaling. So now we will take insulin pathway, and it's a complicated thing. Jada in his lecture spent like 10 minutes saying, he's doing a research in it because it's a very complicated thing. They don't know exactly how does it do its action, how is it related to tumors and these things. So please pay attention because it's very important. Insulin, how does insulin signal? First, let's talk about insulin a little bit. Insulin decrease glucose level, okay? How? By activating GLUT things, GLUT4, GLUT2, or whatever, causing glucose to go from the blood into the muscles or to the cells, which decrease the glucose level in the blood. And also it enhances the usage of glucose to make ATP. So the aim of insulin is to decrease glucose, okay, by increasing its use. If we inhibited insulin, we will have diabetes mellitus, okay? And so now let's talk about its signaling. Insulin signal by which of the following receptors? We took many receptors. Which receptor it used? Insulin used tyrosine kinase receptor. So how does insulin work? By a tyrosine kinase receptor. But the insulin tyrosine kinase is a very special tyrosine kinase. Normally, let's talk about the normal tyrosine kinase, not the insulin one. We have the membrane, we have the receptor, and the receptor is made by two parts, the left part and the right part, and they are not attached to each other. Once the ligand binds, it becomes phosphorylated here and there, and it phosphorylates each other and do its action. That's the normal thing. Let's now talk about the insulin receptor, insulin tyrosine kinase. The insulin tyrosine kinase is special because it's made of 4 subunits. The left part is made by alpha, and it's attached to beta subunits. And the right part is made of another alpha, which is attached to beta. Interestingly, here, these things are not attached to each other. Once the domain, or not the domain, once the dimer bind, they get attached to each other. But the insulin is attached to each other all the time. Now, so it's always in dimerization mode, okay? And made of 4 subunits. Once the insulin binds, now insulin bound to the alpha helical, where does it bind? On the alpha structure, not alpha helical, sorry, the alpha domain. Once the insulin binds to the alpha domain, it became active, and the beta domains phosphorylate each other and activate tyrosine kinase. So the insulin receptor is very special. Why? Because it's made of 4 domains, 2 alpha, and attached to 2 beta. The beta is inside, the alpha is outside. Interestingly, the 2 parts are attached to each other all the time. It doesn't need a dimer to bind or whatever. Once insulin bind, the tyrosine kinase became active on the beta side and phosphorylate each other, and the tyrosine kinase now is active. And since the tyrosine kinase now is active and insulin is out, now we can use insulin to do its action, okay? It's interestingly needed to be said that all chain of the receptor are joined together prior to the hormone binding, so it's always attached to each other. There is no need for dimerization or whatever. It's always a dimer. It has SH2 domain, and now let's talk about its actions before we talk about these things. Let's talk about action. Once insulin become active, it go and increase glucose uptake by increasing GLUT4, 2, 3, or whatever. It increases glycogen synthesis, increase protein synthesis, increase fat synthesis, and increase glucose use to decrease glucose level. Okay, and these are the actions. How do they maintain their action? Is by the insulin pathway. So last time, before we reach to the pathway, insulin bind to the tyrosine kinase receptor. It activates it, and once it's active, it causes many things that cause the GLUT4 or all the GLUT to be active, increase protein synthesis, increase fat synthesis, and whatever. So how does insulin does that? Before I say that, is everything clear? Because now it's very important point. Yes. Okay. So now let's talk about how insulin works. So we have the membrane, we have the alpha, wow, what an alpha? Alpha, beta, beta. Okay. This is the receptor, and it's always bind together. Ligand, what's the ligand? Insulin. Go and bind. Once insulin bind to this, tyrosine kinase became active. Once tyrosine kinase became active, it activates something. Look here, it's tyrosine kinase, okay? Once tyrosine kinase is active, it activates very important thing called insulin receptor substrate, which is IRS. So it's this tyrosine kinase go and phosphorylate something called IRS. IRS is insulin receptor substrate. This is called insulin receptor. So this is a substrate that's related to the insulin receptor. So what's its name? Insulin receptor substrate. So the insulin tyrosine kinase go and phosphorylate insulin receptor substrate, IRS. It's IRS1 actually. And then IRS1 go and activate a very pathway that we talked about, which is the PI3K pathway. PI3K, once it's activated, it activates ACT, as we said. And ACT here in insulin, look here, ACT in insulin doesn't activate bad. No, it go to the GLUT4 inside the cell and make it go to the membrane. So it allow glucose to go to out of the blood into the muscles. So, and that's it. Let's recap. We have insulin binding. Once insulin bind to the receptor, tyrosine kinase become active. Tyrosine kinase activated, it go and phosphorylate IRS1. Once IRS1 is activated, it goes all the way and activates PI3K pathway. PI3K activates something called ACT. ACT go to GLUT4 and make it go and become part of the membrane, allowing glucose to get outside. And that decrease glucose level. And that's the whole insulin pathway. Is it clear? It's very important. So please. Okay. So this is the pathway. Insulin bind, tyrosine kinase is activated, IRS1 is phosphorylated, IRS1 activates PI3K, PI3K activates ACT, ACT causes GLUT4 to become part of the membrane and glucose gets out. So now to the clinical part, the diseases. Okay, it's one slide left, so you'll pay attention. We have one very important disease that is related to insulin, which is diabetes mellitus 2. Diabetes mellitus 2 doesn't happen because we don't have insulin. Actually, we have hyperinsulinemia. So we have very high insulin. So how in diabetes mellitus 2, when we have hyperinsulinemia, we have very high insulin, we have hyperglycemia. Shouldn't insulin decrease the glucose? How in diabetes mellitus 2, when we have insulin, we have very high glucose? Because in diabetes mellitus 2, the cells are not responding to the insulin. So no matter what, no matter how much insulin we have, no matter how much insulin binds to the receptor, the whole signaling pathway is interrupted. So cells are not responding to insulin. And so we have very high glucose. And our body see, oh, we have very high glucose. Let's make more insulin. But this insulin is not working because its cells are not responding because the whole pathway is interrupted. And because of that, we make insulin, so we have hyperinsulinemia, but the cells are not responding, so we have hyperglycemia. So in diabetes mellitus 2, we have insulin, but it can't cause its action because the signaling pathway is interrupted. So there is no response. So we have very high glucose because insulin can decrease glucose. Okay, so in diabetes mellitus, we have hyperinsulinemia and hyperglycemia because insulin is not doing its action. Why insulin is not doing its action? That's because its signaling pathway is interrupted. Okay, so yani it's activating IRS1, for example, but IRS1 is not activating BI3K, or BI3K is not activating ACT, or ACT is not activating GLUT4. So the whole signaling pathway inside is interrupted. So no matter what, no matter how much we make insulin, there is no response because the whole pathway is interrupted. What interrupts this pathway? Three things. First, obesity. Second thing, inflammation. And third thing, we have, actually it's obesity and inflammation in the lecture. I don't remember him mentioning anything other than this. Yeah, so it is obesity and inflammation, or any inflammatory response. These two things cause insulin to be not able to do its action, and it interrupts the whole pathway. How? Okay, let's agree on something. Inflammation has two things playing role, called TNF alpha and interleukin 6. These things you will take in POD next year, so it's new, but just memorize. We have TNF alpha and interleukin 6. These two things are increased in inflammation. What do they do? We said that the insulin receptor is a tyrosine kinase receptor, right? They go and switch the kinase from tyrosine into serine. So now the serine has the kinase. So now the insulin receptor become serine kinase receptor. So now, as I said, TNF alpha and interleukin 6, actually, wait, I want to explain something before that. Or yalla, let's do it. TNF alpha and interleukin 6 go and change tyrosine kinase to serine kinase. How do they do that? By activating something called SOX3, right? SOX3, yeah. So TNF alpha and interleukin 6 in inflammation, they go and activate SOX3. SOX3 go and change tyrosine kinase to serine kinase. Once it becomes serine kinase, when insulin binds, it can't do its action because insulin receptor is a tyrosine kinase. Now we have a serine kinase. So insulin, no matter how much it binds, it's not binding to its receptor because it's binding to a serine kinase. It can't activate it. So once insulin binds, it doesn't do anything. So this whole pathway is interrupted. Even if this tyrosine kinase was able to make IRS1 phosphorylated, it can't activate B13K. It can't activate ACT. It can't activate GLUT4. And this whole thing is interrupted, and that's causing insulin resistance. So no matter how much we have insulin, there is no response, and we have diabetes. So this is the first one, inflammation. Inflammation increase interleukin 6 and TNF alpha. These things go and increase SOX3. SOX3, it changed tyrosine into serine. So now we have serine kinase. So when insulin binds, it activates serine kinase. Even if serine kinase was able to activate IRS1, IRS1 can't activate P13K, it can't activate ACT, it can't activate GLUT4, and so we have insulin. Yeah, so we have insulin resistance, and there is no response. That's the first thing, and that's inflammation. The other thing is obesity. Obesity increases one of the inflammatory responses, which is TNF. So once you eat a lot and you have obesity, your TNF level or TNF alpha level increase. Once it increases, it doesn't activate SOX3 because it's obesity now. It's different than inflammation. Obesity increases TNF alpha. TNF alpha activates something called protein kinase C beta. Okay? So TNF alpha go and activate protein kinase C beta. And this protein kinase C beta go and change tyrosine kinase also to serine kinase. And once we have serine kinase, it's over. We don't have insulin reaction, same to the inflammation. The whole thing is interrupted, and that's it. So, is it clear? Okay, So last time, to recap it, insulin, normally, no, this is the last recap because we just have one slide and that's it. We have the insulin receptor, alpha, beta, alpha, beta. Insulin binds, tyrosine kinase activated, activating something called IRS1, and IRS1 activate what's called, it activates BI3K, BI3K activating ACT, and ACT activating GLUT4, and the other protein synthesis and decrease glucose and these things. Once we have inflammation go and increase TNF alpha and interleukin 6. TNF alpha and interleukin 6 increase SOX3. SOX3 go and change tyrosine kinase into serine kinase. Once we have serine kinase, we can't convert, we can't activate B13K, we can't activate ACT, we can't activate GLUT4, and the whole insulin become meaningless. We don't, it doesn't have any reaction. And so we have insulin resistance and we have diabetes mellitus too. The other thing is obesity. And obesity increased TNF alpha. And so TNF alpha activates something called, obesity activates TNF alpha activate PKC beta. PKC beta also change tyrosine into serine, and serine cause the whole thing also to be interrupted. And so we have insulin resistance. The last slide telling you, you don't need to memorize anything. Literally, he just said this slide is to tell you that we mentioned two pathways we mentioned one pathway of insulin, which is the B13K and ACT and these things. And we mentioned two ways of interrupting it. But in real life, we have many pathways of insulin, and we have many ways of interrupting it, not just inflammation and obesity. We have many ways to interrupt insulin pathway, but also insulin pathway is not only the pathway that we mentioned. There are other pathways and many other things. That's what this slide tells. So you don't need to know anything. It's just that insulin has many pathways other than the one we said. But the one we said is the most important. And we have many ways to interrupt insulin pathway other than obesity and inflammation. But the most important are obesity and inflammation. And that's it. We finished cell signaling 1 and 2. Do you have any questions? Okay, now the last part, because we didn't finish it. We have questions. We will solve these questions fastly. I put the answer in the question just to reduce the timing we take. So we take only 2 hours, similar to what Al-Jadah took. So the first question, adenylyl cyclase activated by a G protein. Which of the following second messenger will be generated? So it's just saying, which of the following is the second messenger in the adenylyl cyclase pathway? And of course, the answer is cyclic A and B because adenylyl cyclase makes cyclic A and B. Simple, right? This is the type of question we have. The second question, manic depressive illness may result from overproduction of IB3 and DAG. And the accompanying signaling process in certain CNS cells, lithium is often useful in treating this illness. Lithium is most likely function to inhibit what? We don't know lithium, we didn't take lithium, but look to the keywords. It's talking about IB3 and DAG. So lithium is actually treating IB3 and DAG pathway. Which pathway is that? Is it adenylyl cyclase, G-alpha-S, phospholipase C, pKa, or tyrosine kinase? Of course, IB3 and DAG are phospholipase C activity because it's the pathway. So the answer is C. Here, a 6-month-old patient present with slight fever, redness, and sneezing, coughing, which is whooping cough. See, this is the keyword. We have whooping cough, which is pertussis. So yeah, and here we have pertussis. And so the question, I will not read the whole thing. Which of the following distribution in cell signaling will result in respiratory tract response to this infection? So it's just saying pertussis is happened due to what? It's all we took that pertussis is due to inhibition of the G inhibitory. So if we inhibit the inhibition, we will have very high stimulation of cyclic A and B because adenylyl cyclase will be overused and we have a lot of cyclic A and B. So the answer is overproduction of cyclic A and B. We have 4 more questions. Which of the following intracellular signal molecules regulate the catalysis of conversion of GTP to cyclic GMP? So just saying what converts GTP to cyclic GMP. And we said nitric oxide, NO, go and activate guanylene cyclase, which convert GTP to cyclic GMP. And so the answer is NO, nitric oxide. Why? Because nitric oxide activates guanylene cyclase convert GTP to cyclic GMP. Then we have the G-alpha S G protein is activated by what? We took that G-alpha S is binding to GDB. Once we remove GDB and bind it to GTP, it becomes active. So the answer is binding to GTP. See, it's very simple recall. Insulin signals via receptors, and it's just saying the insulin receptor is what? The answer is intrinsic tyrosine kinase, because insulin receptor is insulin tyrosine kinase receptor. So insulin signal through what? Through a tyrosine kinase. That's it. And the tyrosine kinase is intrinsic. That's important because, as you remember, the tyrosine kinase is inside, so it is intrinsic. Okay, and the receptor itself is the tyrosine kinase. So C is wrong because it says non-receptor. No, it is a receptor tyrosine kinase because the tyrosine kinase is part of the receptor itself. The SH2 domain of the adapter proteins allows for, like the SH2 is doing what? It's binding the adapter to phosphorylated catalytic receptor. And it's just saying that the SH2 can bind to the receptor tyrosine kinase. I know I'm so fast now, but because I don't want the lecture to be very long. So the SH2 is binding, we use it to bind to the tyrosine kinase. That's it. Binding of the adapter protein, like GBR2, let's say, to the phosphorylated catalytic receptor. STAT is phosphorylated by what? STAT is phosphorylated by the tyrosine kinase or JAK, okay? One of these. So the answer is tyrosine kinase, okay? And if it came to you, put tyrosine kinase, because we said, if there is JAK here, it's also correct, but if there is JAK and tyrosine kinase, choose tyrosine kinase, okay? Because as I said, the membrane has this receptor and JAK go and phosphorylate the receptor. And the receptor phosphorylates STAT because STAT has SH2, okay? So it gets phosphorylated by the tyrosine kinase receptor itself. But also JAK can phosphorylate STAT, but it's a minor effect. So STAT is mainly phosphorylated by the tyrosine kinase receptor. If there is no this choice, please choose JAK because JAK also phosphorylates. And this is the last question. Which of the following plasma membrane receptor activates signaling pathway by forming molecule dimer, see dimer, that result in protein phosphorylation? We said that the tyrosine kinase receptors, not the insulin, no, the tyrosine kinase mainly, okay, is separated. And it needs to bind a dimer molecule. And once it binds this dimer molecule, a dimerization happens. So the two receptors shake hands with each other. And so we have dimerization. So which of the following needs a dimer? The answer is receptor tyrosine kinase. Okay, don't mind this because I was searching for keywords. So that's why it's like this. So the answer is E, receptor tyrosine kinase, because tyrosine kinase need a dimer and need dimerization. And that's it for my part. Now my friend Amjad will explain, by the way, he's one of the best PAL tutors, so I think it will be an easy session. He will explain the steroids and nuclear membrane receptor, and it's an easy lecture. So yeah, that's for my part. And do you have any question? Okay, so